Thank you very much for the opportunity to talk today, and thank you everybody for coming and questions. I'm really excited to talk today about a number of our projects, as I learned how to use the pointer. So really this is a significant collaboration between 3 different labs. We kind of refer to ourselves as the laboratories of host defense and disease. So this is the laboratory of Dr. Amelia Pinto, who has tremendous expertise in T-cell responses, Justin Richner, who's at the University of Illinois of Chicago, who is an expert in mRNA vaccines, and pretty much all 3 of us go back and forth, generating different constructs and coming up with different ideas. But really, how we think about it is my lab and Dr. Pinto's lab work on a lot of emerging pathogens. We use them both to understand pathogenesis and disease. We also use them as big hammers to really kind of understand fundamental processes of innate immune responses as well as T-cell and B-cell responses. And we do that in a number of different animal hosts and mice and non-human primates, but pretty much over the past 5 or 6 years, we've progressed into a lot of phase 1 or helping with a lot of phase 1 clinical trials and now analysis of other human trials. Really, within the context of mRNA vaccines, one of the fundamental questions we're really trying to answer, certainly with this breadth of different viral pathogens, is which is better? A broader immune response, where we have a broad array of multiple of antigens, or a limited immune response that has a very targeted, antigen-specific response with very specific effector functions, okay? And the way we think about it, or the way I try to get my students and postdocs to think about it is, this mRNA delivery system, was really kind of only limited by our imaginations. We're coming this from, a predominantly fundamental system. And one of the main reasons why we've taken this approach is we used to make a lot of infectious clones for RNA viruses, like chikungunya virus or West Nile virus. And 9 times out of 10, we would change a single amino acid to ablate a T-cell, contact residue or B-cell epitope, and the virus would never grow. So we kind of pivoted very early on and just switched them from trying to understand natural pathogens to turning them into vaccines. And that's really kind of what gives us, at least the hypothetical freedom to really kind of understand that. And, an example of that was, very early on in the COVID pandemic, before there were animal models, we used our mRNA delivery system to put human ACE2 into mice, and that allowed us to very quickly identify the T-cell epitopes that allowed, vaccine progress. So today, mostly what I'm going to focus on is flavivirus diseases. So to really kind of think about it, the global incidence of dengue virus disease. So there's 3.9 billion people at risk. And there are approximately 50 to 390 million people each year infected. That leads to 50 to 100 million cases of dengue fever per year. This is a pretty severe disease. This has significant financial impact on a large percentage of the global human population, which results in somewhere between 20 and 50,000 fatalities per year. So here's the particular pathogen I'm talking about. So this is dengue virus. It's a positive stranded mRNA-based virus. So it's like a single open reading frame. Okay. Within the context of this virus, it has 3 structural genes, capsid, PRM, and E. And what you see here is really the envelope protein, which forms as a head-to-tail dimer. When it goes into the structural genes, around the virion, it really encodes both, capsid on the inside, PRM, and E. All right, so we went to go through and start to make a virus-like particle using an mRNA vaccine. We did this originally with DV1. So here we took the PRM gene and envelope gene from DV1. We put it into a mRNA LNP, and we see that we generated VLPs, so virus-like particles, okay? When we're able to immunize mice with this mRNA vaccine, we get very strong neutralizing antibody response with increases over time and doses. This mRNA vaccine is also able to generate a CD8 and CD4 T cell response. Okay? When we take a look at the protective capacity of this vaccine, so we give a lethal dose of DV1, and we can see 100% protection. When we take these vaccinated animals and we do a passive transfer of serum, we see that within a homologous challenge, meaning vaccinating against DV1, pulling serum after mRNA vaccination and passively transferring it to animals and rechallenging, that we get very strong protection, from the polyclonal antibody response. Okay, but for dengue virus, depending upon how you think about it, either it's a diversity problem, so if you're coming at it from a vaccine perspective, all right, there are 4 serotypes of dengue virus, and they differ at the amino acid level of somewhere between 25 and 40%. To make it even more complicated, although I'm not really going to drill down into that today, each individual serotype is composed of multiple genotypes that can even vary, up to 7% between these genotypes. Another way to put this in perspective is West Nile virus, another flavivirus, and Japanese encephalitis virus are actually more closely genetically related than any 2 dengue serotypes. So these are really about 4 different kind of flaviviruses that cause very human similar diseases, but amino acid sequence-wise, they're pretty quite diverse. Okay? The other aspect that's both really exciting within the context of flaviviruses from a basic science field, but very nerve-wracking from a vaccine development field, is that flaviviruses, which there are about 72 that can infect the human population, that these are not geographically isolated. These are all overlapping. There is a high, potential for individuals to be infected with or be exposed with over their lifetime many of these flaviviruses. So we're not dealing with one vaccine for one virus. We're dealing with how do we generate vaccines which can cover so many of these different viral pathogens. Okay, so then we have this third extremely exciting or interesting phenomenon. It's called antibody-dependent enhancement. Okay, and what I'm going to talk about today is the data not only that we have from animal models on antibody-dependent enhancement, which I think really gets at mechanism, but also really the real-world human data on antibody-dependent enhancement. So to explain what this is, so similar to the DV1 vaccine that I walked you through the data with in the beginning, is if an individual gets bit by a mosquito that has DV1, you generate an immune response to DV1. That immune response will eventually help you clear that DV1 infection, and then it is highly unlikely that you develop a DV1 disease again. But remember, there are 4 different serotypes of dengue virus. So what happens in a certain percentage of the population is upon second dengue virus infection with a different serotype, okay, now you have an antibody response to DV1, okay, that has the ability to bind a different serotype of dengue virus. But what we know mechanistically is usually that isn't a neutralizing antibody response. There's just enough antibody for it to bind that what winds up happening is you get antibody-dependent enhancement. So the antibody binds the virus. Cells that uptake through Fc receptor that virus can become infected. Because with these flaviviruses, dengue virus replicates preferentially in cells that naturally express Fc receptor. So it's a second way for viruses to infect cells. That winds up increasing the number of infected cells upon infection, and that leads to a higher level of viremia and an increased disease. So we've seen this in real life when it comes to rollout of vaccines. Yesterday we talked about the yellow fever 17D vaccine. This is the platform that DengVax was originally generated with. So in this particular vaccine, what they did is they took PRM and E and they replaced the yellow fever PRM and E. They mixed those in a ratio and they vaccinated individuals. This was eventually halted. Now it is back into use, but the WHO has changed how this vaccine is being rolled out. So it's being rolled out in areas where there is at least 50% of the population is dengue virus positive, and it's being rolled out to individuals who are 12 and older. And it's being done that way because you really need to essentially boost or use this vaccine to boost pre-existing immunity. If you are dengue virus naive, like you are with young children, there was a safety signal where it was increasing severity of disease upon primary dengue virus infection. So, we have the situation where we have antibody-dependent enhancement. So graduate students in the lab were really excited about the work and the development of our mRNAs and the fact that we're able to really kind of play with the system. So Mariah Hassler, a graduate student in the lab, who now is a postdoc in John Hardy's lab, she developed a whole series of different animal models. So this gave us, in the context of natural infection, a mechanism to understand, prevention of disease or increased severity of disease. And she did it with a whole panel of different flaviviruses. So West Nile, JEV, Usutu virus, Zika virus, and a whole bunch of other flaviviruses that we don't commonly talk about. So using this system, we just asked, in the context of natural infection, how does prior flavivirus exposure in a lethal challenge model to Zika, what's protective, what's not? So what she did is she immunized a huge number of animals with either Zika or DV2 or DV3 or yellow fever. Okay, we bled to take a look at, what was the immune system looking like, T-cell responses, antibody responses. Okay, and then 30 to 60 days later, she came in with a lethal challenge of Zika. And she wanted to ask, how do these different viral infections protect against Zika? So if you have a prior Zika infection, it protects you against Zika infection. Okay, where we see also, pretty good protection or protection against lethal Zika by DV2. We see limited protection from when you have a prior yellow fever or DV3 infection. So, and this is true not only of mortality, but this is true of disease symptomology. So this kind of more clearly points out the differences in the potential protection in the context of natural infection. So she went on to take a look at the neutralizing antibody response. And what you can see here is really mice don't generate a robust cross-neutralizing antibody response that's similar to what you see in humans after approximately 3 to 6 months post-primary infection. But upon Zika challenge, you have a rapid onset of neutralizing antibody response. Okay, she turned around and, like I said, we took a look at germinal center B cell populations, CD8 T cell responses, CD4 T cell responses, and what she identified using the animal model is, cross-reactive CD8 T cells were one of the best predictors of protection. So the larger number of cross-reactive CD8 T cells you have, the greater reduction in viral load you have. Okay, so that came, to another student, Alex Dixon, who has really become our mRNA vaccine guru in the lab. And we call this vaccine, the dengue T-cell vaccine. So, and this is really where we're able to kind of play with molecular biology and do some really exciting things. So this is our DV1 vaccine that I talked to you about the first few slides. And what we've simply done here, we, meaning Alex, put a P2A, so it's a single mRNA open reading frame. We have the DV1 VLP, and it's followed by a single cross-reactive CD8 T-cell epitope. And we do this just using, an old school MHC class 1 loading approach using the E319 from adenovirus that was used in all these DNA mini genes by John Udell and many others. We vaccinate these animals, whether the cross-reactive epitope is derived from the Zika sequence or the dengue sequence, and we vaccinate these animals twice, we see a pretty good CD8 T-cell response. Okay, so then we went to take a look and we say, hey, if we vaccinate using this dengue mRNA vaccine, what does it look like in this heterologous challenge when we challenge with Zika virus? So these animals were vaccinated twice. Then they were given a lethal challenge with Zika virus. So if we vaccinate with EGFP, we see no protection. If we vaccinate with the dengue virus VLP, this DV1 protective vaccine, we see almost no protection. About 5 or 10% of the animals are vaccinated. If we take the CD8 T cell cross-reactive epitope, We see a little bit of protection against Zika virus, but not much. But when we combine the DV1 VLP vaccine with this cross-reactive CD8 T cell response, we're able to really boost and protect in this heterologous fashion. And I think this really kind of shows the advantages of mRNA vaccines, where we can plug and play and put different type of epitopes and different things into the vaccine. So just to summarize this first part of my talk, so mRNA vaccines expressing PRM and envelope of flaviviruses result in the formation of virus-like particles. mRNAs generate VLPs and protection against homologous flaviviruses, but they're unable to protect so far against heterologous flaviviruses in our animal models. mRNA vaccines can be adapted to express antigens in a number of different ways, allowing inclusion and exclusion of different antigens. And inclusion of cross-reactive T-cell epitopes into mRNA vaccines that already generate VLPs increase cross-protection. So now I'm going to pivot to mRNA vaccines to CoV-2, where to us, it allowed us to start to use the same technology and same approaches to kind of address very similar questions. So rather than thinking of all of these different viral variants, as almost like different serotypes of flaviviruses, where we really wanted to understand how can we add and subtract different immunological components to better cross-protect. So in this particular case, as we now have these Omicron variants, we developed a new strain of K18 mice. They're pretty much K18 homozygote mice. Other people in the lab wanted to ask questions of, how does the level of human ACE2 expression affect vaccine protection? So we know that human ACE2 expression varies in the human population. When you develop animal models that have varied human ACE2 expression levels, how does that affect, your ability to protect? And how does that affect transmission? These are two questions that are still ongoing in the lab. I'm not going to address them now, but this animal model certainly does allow us to start addressing that question. So just kind of validating this initial model. So if we take a look at Omicron BA1, which we know in the standard K18 model causes little to no disease. As far as severity, you can measure viral output, but not disease. So in these K18 mice, they rapidly succumb upon BA1 infection. So at the time, people have been playing with different stabilizations of spike. At this particular time, the addition of multiple prolines is kind of really where the field was going. So this is just our starting point. We use the S5P as our spike backbone. And so when we take this mRNA S5P vaccine and we use it in just the K18 model, whether we have like a WA1 type challenge or we have a Delta challenge, we see that we can protect with just the standard S5P or spike vaccine. Okay, but how do we use different antigens and how can we use this mRNA technology to really kind of outcompete the type of variants or at least antigenic diversity that we see in viruses circulating? So our next step was to start to generate CoV-2 VLPs using an mRNA vaccine. Right now, I have to tell you that we're still validating a lot of the expression. Are we really formulating VLPs? What are the percentage of VLPs? So we still have a lot of questions, but I did want to share a lot of the preliminary data we have. So here's just a very simple experiment with our K18 homozygote model. So really, we wanted to compare if we have the S5P vaccine and we challenge with Omicron, or this S5P plus this addition of M, E, and N as additional vaccine antigens, can we increase our ability to protect? And that is exactly what we saw. Like I said, I just want to repeat, we're not sure yet as to if we're really getting true VLPs. So this right now is just to the point of increasing the number of antigens delivered, expanding the immune response, and seeing an increase in protection using this K18 model. So Once again, I am just one person here sharing everybody's data. We have fantastic collaborators. The Pinto Lab is a fantastic collaborator. We've had a lot of support from the NIH and other groups, and I've had great mentors, and thank you very much for your time.